بسم الله الرحمن الرحيم. هنتكلم النهاردة بإذن الله عن blood hemolysis. Content of the practical session. Number 1, blood hemolysis definition and causes. Number 2, different blood hemolysis sample. Learning outcome. Identify concept of blood hemolysis and causes of it. Demonstrate and differentiate samples of non-hemolyzed and hemolyzed blood. Demonstrate different hemolyzed blood samples by different causes. Discuss mechanism of hemolysis of hemolyzed blood samples by different causes. Learning outcome number 1. Identify concept of blood hemolysis and causes of it. Blood constituents. Blood contain 45% of cell and 55% of plasma. This is the picture of RBCs, platelet, white blood cells, and the blood vessel. Red blood corpuscle. Erythrocytes are biconcave non-nucleated cells consisting of a special membrane around a concentrated solution of hemoglobin. This membrane composed of a mixture of proteins and lipids such as semi-permeable membrane with great selective properties. Blood hemolysis definition. Blood hemolysis is rupturing of RBCs and passage of hemoglobin to the outer medium, and this is called blood hemolysis. This is the picture of normal RBCs, spherocytes, which are spherical in shape, and ruptured erythrocytes, release of content into blood plasma. Causes of blood hemolysis. Hemolysis in vivo, which may be due to extrinsic factors, APO-RH incompatibility, or intrinsic factors as red cell membrane defect, abnormal hemoglobin, or enzyme defect. Hemolysis in vitro, in the lab, improper technique during collection, incorrect needle size, excessive suction. Number 2, change in osmotic pressure, like placing RBCs in hypotonic solution. Number 3, dissolution of lipid part of the red cell membrane by fat solvent or saponin. Number 4, denaturation of the protein part of red cell membrane by acid or alkali. Learning outcome number 2, demonstrate different samples of non-hemolyzed and hemolyzed blood. Hemolyzed and non-hemolyzed blood. Difference between hemolyzed and non-hemolyzed blood samples. Hemolyzed blood samples solution is transparent, as hemoglobin solution in water is clear. Non-hemolyzed blood samples solution is opaque or turbid, as RBCs are suspended in solution. This is the difference between hemolyzed and non-hemolyzed blood sample. Non-hemolyzed blood sample are turbid, but hemolyzed blood sample clear or transparent. This is also picture for hemolyzed and non-hemolyzed blood sample. Note that hemolyzed samples is transparent because there are no cells to scatter light. Learning outcome 3 and 4 demonstrate different hemolyzed blood samples by different causes and discuss mechanism of hemolysis of hemolyzed blood samples by different causes. Effect of osmotic pressure. Osmosis. Water flows across semi-permeable membrane into the solution with a higher solute concentration until the concentration of both solutions are equal. The red cells plasma surrounding them. This osmotic pressure is also equivalent to 9% NaCl solution, which is postulated. Method. Prepare 3 test tubes. Number 1. Then add 3 drops of defibrinated blood and mix. Tube number 1, 5 ml plus 3 drops defibrinated blood. Tube number 2, drops defibrinated blood. Tube number 3, 5 millisaccharose plus 3 drops. Wait 30 minutes. Observe. Observation. Tube number 1 is isotonic, which contain 0.9% NaCl. Tube number 2 is Tube number 3 hypotonic, which contain distilled water. Tube number 2 no hemolysis. Tube number 3 mechanism, which is hypertonic. Water go out from the red blood in shrinkage or pronation of the RBC. Tube number, because osmotic pressure is equal in and out the RBC. So swelling and rupture. This is also picture for effect of osmotic pressure. Now, in picture A, RBCs immersed in isotonic solution. B, RBCs immersed in hypertonic solution. C, RBCs immersed in hypertension. What would you expect to observe under microscope after 30 minutes from RBC suspension in a solution which is hypotonic to RBC? Answer. No cells would be visible as RBCs would be hemolyzed, making blood transparent. Factors affecting lipid of tube number 4 and number 5 containing the following. Tube number 4, 5 ml defibrinated blood plus 2 milliliters defibrinated blood plus 2 drops saponin. Mixed by in. Wait 30 minutes. Observe whether hemolysis is taken. Observation and mechanism occurs as ether is fat solvent, so it will leave cell membrane. Tube number 5, hemolysis occurs also as saponin will lower the surface of lipid part of membrane of red cell and causing its disintegration. Pyrolysis causes hemolysis by the same mechanism through disintegration. Factors affecting protein of the red cell tube number 6, 7, 8 containing the following. Tube numbers defibrinated blood plus 1 milli or 0.1 normal 7, 5 ml saline plus 3 drops defibrinated blood or 0.1 normal NaOH. Tube number 8, 5 ml saline plus 3 drops defibrinated blood 50 to 70 degrees centigrade. Wait 30 minutes. Observe whether hemolysis has taken place or not. Observation and mechanism. Hemolysis occurs due to denaturating part of red cell membrane, leading to loss of its normal permeability of hemoglobin to the outside. Formation of acid or alkali hematine, which means that acid or alkali bind to heme in these test tubes will give the 8. Hemolysis occurs by the stated hemoglobin by atmospheric oxygen. Ferrous ion become ferrous. Summary: hypotonic solution or hypertonic solution. Isotonic solution like percent NaCl, which is isotonic solution, which hypotonic solution 10 ml distilled water plus 3 tonic 0.9% canned sugar plus 3 drops of blood. Observation: hemolysis occurs only in the hypotonic solution. Comment: isot and inside RBCs, so no hemolysis occurs. Hypotonic water passes into the RBCs to equalize RBC. Hypertonic solution shrinkage of RBC. Effect of emulsifying agent and fat solvent. Agent like saponin, fat solvent like ether. In emulsifying agent, saponin is emulsifying agent which causes emulsification of the fat by lowering surface tension between fat and water. Saponin binds with cholesterol in lipoprotein stroma, break up lipid protein complex of stroma. In fat solvent, ether is a fat solvent, so it will lead to dissolution of the lipid part of red cell wall and rupture RBC. Effect of protein denaturing agent. Protein denaturing agent like acid, alkali, or heat. In acid, we put 1 milli of 0.1 normal HCl. Alkali 1 milli of 0.1 normal NaOH. In heat or physical agent, we put the sample in warm water bath. Hemolysis occurs in all the tubes. In acid and alkali, denaturation of the protein part of RBCs and the formation of acid or alkaline hematine, which gives a brown color. In heat, denaturation of the protein part of RBC's membrane, causing hemolysis, and autooxidation of the liberated denatured hemoglobin by atmospheric oxygen, so give the brown color. Thank you too much.